we saw that we could use Biopython to parse FASTA files. And now we're going to see another format, which is quite useful and important, the GenBank format, which we can take, for example, from UniProt. So let's see an example. So let's go to the link that you see here and go to that URL. So this will straightforwardly bring me to the page of the BRCA1, one of its transcripts in UniProt, in RefSeq for human. And this is exactly like the RefSeq pages that Michal showed you last week. And if we want to download that, we can press the Send To button that you see here on the top right of the screen. And then we can choose to save it as a file. And then we can choose from a bunch of formats. So the default is a GenBank full, which we're going to download now. But you can also choose other formats, including the FASTA, if you just want the sequence. So if you just want the sequence of the transcript, you'll use the FASTA file, which will give you the sequence with just maybe its ID and description. But if you're interested in all of the annotations, like you see here on the page, you will choose the GenBank full format. And then we can create a file and save it. Sorry?
Could you do it again with the send to? I missed the part.
You press Send To, File, and GenBank full. Yeah, that's the default, should be.
And then create file.
Yeah. OK, so here we see it. Instead of the FASTA suffix, and now we have the GB extension. And let's look at this file and see what it looks like. So if we open it with Notepad++, you can see that it's exactly the same format that you see on the RefSeq page. So basically a textual format that contains all of the annotations. Now it's a textual format, so if you wanted, you could parse it on your own. But it would probably be somewhat a laborious and annoying task, given the complication of this format. Luckily for us, we have Biopython to the rescue, which knows how to parse these kinds of files. So if we open this file and then parse it with a SeqIO module, this time we specify the GenBank instead of the FASTA format. Then it knows just how to read it, and we get the record objects. By the way, do you know why we have this comma here, which is extremely important? OK, what does SeqIO parse return? What is the value that you get from SeqIO parse? A generator, right? So if I didn't use this comma, what would record be? It would be a generator of this file. But when I use the comma, what happens? Sorry? Exactly. When I use this comma, it knows that I have a generator, and I expect this generator to have just one element. And I want that element to be stored into this record object. If it had two objects, I could use maybe record 1, record 2, and extract each one of them into a different variable. But of course, if I did that now, it wouldn't work because I have just one element. So when I use this comma, it's a way to specify to Biopython that I know that there should be just one sequence, and I want the record, not the generator, into this variable. Okay? So I do not want to work with a generator. I just want the actual content. And now it will actually read it and give it to me. So like I said to you in the first lesson, note those little commas. They are extremely important, and they are very easy to overlook. OK, now let's see what I get. So you see that the type is, again, the sec record. It's the exact same type that I got when I parsed the FASTA file. And that's the same type that I get now when I parse a GenBank file. Okay, so that's the same type, but it contains somewhat different details. Like with FASTA files, that we still have the ID and the description, and you can see that the ID it takes is essentially, I think, the excision, maybe with the version. And the description that you see here, I think it takes it from the definition. So it knows where to take the ID and description from. Okay, not like with the FASTA files. Now it's a different format. And again, like with the FASTA format, I can just go to the sequence, and it will give me the sequence, which appears at the very end of the GenBank format. You see it here. I didn't know how to parse it properly, so I will get it in uppercase and without all of those numbers and without the white spaces. So it knows how to parse just the actual sequence, which is an RNA, which is represented like a DNA with T's instead of U's. And OK, so up until now, it's like the SEQ record that we worked with FASTA files. But now on top of that, we have all the annotations that appear in the GenBank format. So for example, we can access the record annotations, and we get some kind of dictionaries. And if I look at the keys of this dictionary, you can see that it has all kinds of things. It has molecule type, topology, and so on and so forth. Basically, all the annotations that appear here. So everything becomes a key-value pairing and put into a dictionary that I can access. And for example, if I want just the taxonomy, you see that I get a nice list of all the taxonomy, starting from Eukaryotia all the way down to the genus of the Homo sapiens, which is Homo. And you see that it really parses it really nicely. Here I have the organism, and below it I have the taxonomy with some kind of format separated by semicolons. But when I use Biopython to parse it, it knows how to parse it and just give it to me as a list of strings. So it does a lot of parsing for us, and we do not need to worry about this parsing. Now, additional to the annotations, we also have the features, which is the next part that appears in this format. You can see that before the sequence, we have the features part, which specifies different features. So unlike the annotations that specify things about the transcript as a whole, the features are some kinds of more specific annotations that are related to specific parts of the sequence. So for example, we have an exon feature, which is between position 1 to 213. And then on top of that, we have all kinds of additional key-value pairing, which is known as the qualifiers. So if I just take the first three features, you can see that each one is printed in a nice textual way, and we can see the type of the features and its locations and all the different qualifiers. And we can also access those different fields specifically. For example, let's, as an example, say that we are interested in the exon positions of this transcript. So we can iterate through all the features, and then we get a type of each feature, and we compare it to an exon. And if it is an exon, we can go to feature location start and feature location end, and convert them to integers. There are different types, I think, by default. What is it? Yeah, it's in some exact position, but we can just convert it to an integer, and then we can print all the exons, and you can see the coordinates of all the exons in this transcript. Okay, so that's just one example, but again, if you want to do other kinds of analysis, you can do that, and you can also access the qualifiers through this Python interface, and you will see example of that in the exercise. So it really gives you access to everything that appears in this record. Now, if we want, we can also convert it to a FASTA file. So to do that, we can simply open a new FASTA file in writing mode. And then we can use the SeqIO write to give this one record as a list of one record. give it a file and state that we want it in a FASTA format. And when we do that, we will get the output to FASTA file in C downloads that I created. And here it is. And you can see that I get the essential same sequence, but just with the sequence and the excision and the description, and nothing else. So even though this record object was read from a GenBank format, we can use the same SeqIO module to write it as a FASTA format, because it also contains the same kinds of fields and all the different annotations that also appear in the GenBank format are just ignored when you try to save it as a FASTA file. So it is very compatible, and there is a reason to use the same object to represent different kind of records that come from different formats. So like I said at the beginning, there are many different formats that are supported by the SeqIO parse function. And we just covered 2 of them, but there are many more, like, for example, the FastQ format that I suppose you know, that we get from next generation sequencing, and also the Uniprot XML, which Michal mentioned today. So if you download a Uniprot record in an XML format, you can use the SeqIO module to parse it, and also PDB formats. But I should note that the SeqIO only deals with sequences. So if you use it to parse a PDB sequence, you will just get the sequence and not the structure, which is the main thing that you find in PDB files. If you're actually interested in the sequence, you can use a different module, which is the Biopython, which can also parse the structure, and then you can get access to the exact position of each atom in the protein and stuff like that. And this is a partial list. You should go to the documentation if you want a complete list of everything it handles. OK, 2 more quick and small features I wanted to mention. The first thing is a CodonTable. So in previous lessons, if we wanted access to the CodonTable, we needed to hard code it into our code. But Biopython already provides it for us. We can import from the CodonTable the standard DNA table, which is used by most organisms. And if we print it, we get a very nice textual representation, which is much more similar to the kind of tables that you will see in textbooks. And if we want to actually use it, we can, from the CodonTable, we can choose the forward table attribute, which will give us just a dictionary, mapping each codon to its amino acid. So for example, if we want to translate the TTG codon, we can access this dictionary in that key, and we will get that this is a leucine codon. But it only contains amino acid codons. If you want the stop codons, you will need to get it from a different list. You have the stop codons, which just gives you the list of the 3 stop codons in the standard CodonTable. Another very small thing that Biopython gives you is the GC content. If you want, you can just use this GC function from SeqIO. Again, this SeqIO has many more functions, which you should probably check out at some point. But this is just one small thing I wanted to mention. OK, now a final thing I wanted to talk about with respect to Biopython. This is also related to other formats that we saw. So let's begin with an example. So suppose I now have a FASTA content, but instead of reading it from a file, I get it as a string. Maybe I got it from somewhere else. Maybe it was just part of a file that I parsed, or maybe I got it from directly from some website into Python. And I do not have a file on the operating system file system with the content of this FASTA. I just have the content in memory as a string. And now suppose that I want to use SeqIO to parse it. OK, so I use SeqIO parse, and I give this FASTA string, and I specify I want it as a FASTA format. And what happens? I get an operating system error. And the reason that I get this error is because, like I mentioned at the beginning of this lesson, when SeqIO parse gets the first argument to be a string, it assumes that the string is a path of a file containing the FASTA file, not the FASTA file, not the content itself of the FASTA file. So I'm in somewhat of a trap here because I don't really have a way to give the actual content of the file to the parse. I can only give it some file. It can be a file handler or a path, but the SeqIO parse expects to get an actual file. Now, I can, in principle, if I wanted to, I could create a new file, a temporary file. I could write down the content of this string to this file, and then use that file and to feed it into the SeqIO parse function. But I argue that this is probably not a good solution. Can you think of reasons why it wouldn't be a good idea to just use temporary files and why I would prefer to avoid it and work with the content that I already have in memory? OK, yeah, it's slower. Why is it slower? Yeah, so basically storage operation, I/O operations are generally much slower than internal CPU and memory operations, which are much faster. Okay, it stems from the fact that the memory card in your CPU is very close to the actual CPU processor, and they communicate very fast with one another. And it also has very quick random access to the CPU. Whereas when you read from the storage, even it's a fast storage, like an SSD card or something like that, it's much more remote from the CPU, and it's much slower to actually read from the storage. So that's one reason we want to avoid using the storage. Can you think of more reasons? OK, so there are a bunch of reasons. For example, I might want this program to run not only on my computer, but on other people's computer. And this is somewhat rude. When somebody runs your program, you do not want to spoil their storage with some arbitrary files without them knowing that. So when you use some package that you download, when someone gives you, you do not expect it to start writing things to your storage, to your file system. And in many cases, you might not even have permission, so you do not even know what is the directory structure of the computer that you work on. Now, virtual environment will let you, it's something internal to Python. So it's something that you install to create a virtual environment of Python itself. It has nothing to do with other files, data files, that Python uses. And also, when you start working with temporary files, you can very often get to all kinds of problems. For example, your program can terminate in the middle, and you forget to delete the temporary files, and you end up just spoiling the disk with many files that you didn't even intend to create. And many times, you can have some conflicts. So maybe if you run a few instances of the same program, they will try to read and write to the same file. And maybe some of them will fail, and you'll have to start handling all of those edge cases and make sure that the file names are distinct. So it's a complete headache to start working with files, and you want to avoid it. When you can, working with memory is just a lot easier and a lot faster. And you should do that always, instead of working with files. Sorry, I didn't get it.
Sometimes it's better to use a temporary file if you save a really big variable in your memory.
Yeah, OK. So yeah, sometimes if you have memory constraints and you want to do some operation that doesn't even fit into your memory, you don't have a choice, and you need to use files. It's right, yeah. But if you can avoid it, I think you definitely should avoid it. So you need to use the file system only if you must, if there is a reason for that. If you want to keep it, you want to store this data for later, or you don't have enough memory, or reasons like that. These are good reasons to use the files. But just to bypass some constraints of some package, I will now show you that you can do it in a much more elegant way that doesn't really require you to use actual files on your disk. So to do that, we can use a new type, which is called a StringIO, which you can import from the built-in IO package. And what it basically does, it creates you a file-like object. So it's very similar to the file handler that you get when you open a file, only it doesn't represent a real file. It represents just some, you can think about it like virtual, a file that is just in the memory of the program. So we do that by creating a StringIO with the fast string as the content of this like virtual file that we want to create. And we store it into an f variable. And you can see that f is just a StringIO object. But like a file handler, it has the exact same interface. So it has a read function. And we can use that to read the content of what we put within this StringIO, which again, I remind you, is just in memory. It doesn't point to any real file. But the nice thing about it, since it gives us something that imitates a file handler, we can give it to any function that expects to get a file handler. And in particular, we can give it to SeqIO parse. If we give it a StringIO wrapping of this fast string, now we can actually read the content and parse it into FASTA. OK, so this is a nice trick. And by the way, the StringIO, we can not only read from it, we can also write to it. So we can create an empty StringIO, and then we write some text to it, and then we can read it and get the thing that we have written. By the way, do you know why we need the seq here? Yeah, exactly. So a file always contains a pointer to its current location. And when you write to a file, it will always point to where you stopped writing. So the next time that you write, it will continue from the end. So if I didn't have this line, I would just get an empty string, because I would already be at the end of the virtual file. So I need to reset its location back to the beginning, so when I read it, I get everything again.
It doesn't overwrite when you write over a file? The W?
Yeah, so if you write something, yeah, I mean, if you open an actual file and write on it, it will overwrite it. But in this case, it's just a StringIO, which is empty. I didn't put anything in it. So yeah, so when I write to it, it just writes to an empty quote-unquote file. Now, this applies not only to the SeqIO parse function, it also applies, for example, to the CSV module that we saw in the previous lesson. So the CSV module has the exact same problem. It expects to get file handlers, and it doesn't know how to work with just strings in memory. So for example, if we want to turn, say, a list of lines here, that we have each line as 3 values, and we want to get a string representation, a string of the CSV representation of this data, normally we would have to go through some file. But here again, we can just initialize an empty StringIO, and when we create a CSV writer, we just give this empty StringIO, and now we can write all the rows of the CSV writer using the interface that we know. And now, again, we go to the beginning, we reset its location to the beginning, and we read what we have. We will get the string representation, the CSV representation of this content that we wanted. Any questions? OK. So that summarizes the topic of Biopython. Now, the next small topic that we covered today, this will be a continuation of the functional programming that we started to cover last week. So let's continue from where we stopped last week and do a quick recap of some things related to one-liners and functional programming that we learned. So let's begin with the demonstration of working with the VIPERdb dataset that you worked with in the previous, I think it was the lab exercise. So you can download it again from the Moodle if you haven't already. So I opened this CSV file here, creating this CSV reader from it. And now what I do, I get the first line using the next function. I will get the first line of the CSV, which in this case, since it's a table, I will just get the headers of all the different columns. Okay, so basically the column names is the first line in the CSV. And now what you see here, I use some kind of list comprehension to parse the rest of the files, but instead of just getting them as lists, like CSV Reader gives me, I want to turn them into something which is more convenient to work with. I want to make them into dictionaries that map the column name to the value of each line. So the way I do that, I use a list comprehension, like we learned. So for each line in the CSV Reader, so remember that each line is just a list of strings. And what we do with zip, we use the zip function, which we learned again in the previous lesson. We zip the headers of the VIPERdb, which we parsed earlier, to the line content. And what this will give us, it will give us a list of tuple pairs. And each pair will be the two matching values, one from the headers and one from the line. And since that they are ordered in the same way, we will get each one to be the header followed by the value. And then if we take this list of pairs and turn it into a dictionary, we will get a dictionary that maps each header name to the corresponding value in that line. So you can see, for example, here, if I take the first 5 records that I parse in this way, I will get that the first record has an entry ID of this and that, and a family like that. And you can see that I have the mapping that I wanted of each header mapping to the value of the first record, and then the second record, and so on. And then I can do all kinds of analysis with it, and I can work in a more comfortable way than I would have if I needed to just work with lists of strings and starting to work with indices. So for example, I can iterate through all the records and get the outer radius, which is just a string. This is what CSV Reader gives me. But I can convert it into an integer, and then I can sum over it and divide it by the number of records, and I will get the average outer radius of all of these records. Okay, so this is just a recap, illustrating again some of the capabilities of Python that we learned in the previous week. And now let's try to cover some more ground. So first off, I want to show you the iteMgetter function of the operator module. So the operator module, which provides us some nice interface for all kinds of Python operators, has this function which is called an iteMgetter, which what it does, if we create an iteMgetter of something, it will just create as a function that applies this something using the square bracket notation on whatever we give it. So for example, if we take an iteMgetter of 1, this will use the square bracket 1 on everything we give to it. Namely, it will give us the second item if we use it on a list or something else. So for example, if we give it a string with 3 letters, and we invoke this function that we get on this string, that we will get the second letter, because this is what this function does. Okay, now this is really equivalent to just creating a function using the lambda syntax that we learned last week. So let's see an example. So let's say I have a list of pairs, and I want to sort them by the second value, not the first, which is the default of sorting. So to do that, I can just create a lambda function like we saw in the previous lesson. I can create a function that for each pair will give me the first item. Or I can just use this item getter that I created earlier, and it will do the same thing. This is actually just a shortcut to this lambda. So item getter 1 is just a shortcut to creating a lambda function mapping everything to that thing with square bracket 1. Okay, and it works not only with lists, it also works with dictionaries. So again, an example, let's say I want to take all the VIPERdb records that I created, and I want to sort them by the genome. So I can create a lambda function mapping each record to the genome of this record, or I can just create an item getter that will get the genome as the item that it will get, and this will create this function, which I use here as the key of the sorting, and I will get the same list. Okay, so this is just a shortcut to use this square brackets operator, and I will say more generally that the operator modules, it contains pretty much a functional version of everything that you have in the Python syntax. So for example, if you want to create a function that for each 2 elements will check whether the first is lower than the second, you can use the operator LT function, and it will give you that function. Okay, any questions? Okay, let's see some more useful modules that we may want to use. So we have the itertools module, which contains a lot of useful kinds of functions to dealing with iterables, or things we want to iterate. And we'll see here some examples. We'll see the repeat, change, cycle, product, and so on. So let's import those. So let's start with repeat. Repeat is very simple. We just give it some element, and we give it a number of times. And this will create a generator that will give us this element the number of times will be specified. So you can see that it's actually a generator. It doesn't really create everything in advance. But if we call the next function, we'll just get an A. And if we want everything, we can convert it to a list. And you can see that indeed we get A 10 times. Okay, so this is sometimes useful. Another sometimes useful function is the chain function, which given some number of lists, or more generally of iterables, it will just chain them together and again create some generator that we can then turn into a list, and it will concatenate all of those together, and we'll just get one long list with everything together. We also have the cycle function, which given some collection of items, will just cycle through them forever. Okay, so this will actually create an infinite generator that will never end. So if we just do a for loop on it, it will just continue forever and it will never stop. But for demonstration, let's just run for maybe 10 iterations. And you can see that each time that I call the next function, at the beginning it will just give me the 3 items. And then on the 4th iteration, I will get the first item back again, and it will cycle through them on and on forever for as long as I keep asking for the next item. There is also the product function, which given, say in this case, given 2 sequences, it will give me the Cartesian product of these, namely all of the combinations to take one element from the first group and the second element from the second group. So given groups of 2 elements each, there are 4 possible combinations. So we'll get A and C, A and D, B and C, and B and D, all the different combinations. Now it doesn't work only with pairs, I can also use it with more groups. So for example, if I get all the possible bases and I give it 3 times, it will basically give me all the triplets of bases. Okay. Now, by the way, I wanted to mention that I use the variable name bases with an uppercase and not with lowercase because this is a convention in Python, and I encourage you to stick to this convention. When you use a constant variable, namely a constant variable that you just hard code at one place and then you never change it, then the convention is to use uppercase notation to indicate that this is a constant. This is not something that should change across the flow of the program. Of course, Python will not enforce it, so it is like any other variable, but it's a good way to indicate that we use some hard coding of some value, that we just want to keep it as it is. OK, so back to the flow. So now that we use the product to get the triplets of letters, we can use the join function to turn them into strings, and we can get all the possible codons, just the triplets of letters, using this product function. A similar function, but somewhat different, is the permutation function, which, given some iterable object and then a number, will give us all the possible permutation of this. So a permutation is not like the product, because a permutation doesn't allow duplication. So it will just give us all the possible values, all the possible options, to get 3 values which are different from one another. OK, so I will not get something like AAT, only 3 letters that are different from one another.
So just samples from one of the elements that you want all the time, it samples only once. So I sampled A, I sampled C, I sampled T, and that's about it. You cannot sample like twice as A, twice or T.
Yeah, exactly. Yeah. Yeah. And the final function I wanted to show you is the group by function, which, given again some list or collection or any iterable, and given some function, which is used as a key here, it will use this function to evaluate, to get a value, from each element in the sequence, and then it will group all the values that have the same key. So for example, if I give it a function that, given a codon, will get you just the first 2 elements, you can see that it will give me groups, and each group will have the same 2-letter prefix. Okay? So it will group them by the 2 first letters and allow the 3rd letter to change. So what it will give me, again, it will create a generator of pairs. Each time the value that this function gives me, that is used for the grouping, and then the actual elements of this group. And in this case, the grouping is by the prefix, and the group will contain the actual codons. You can see that I can print it and get, for each 2 letters, all the codons that have these values. Now, it's extremely important to state that this group by function is like everything in itertools. They create it to be lazy. So it doesn't do any calculation in advance. It just, like any good generator, it will only do the work upon demand. So on any iteration, it will just give you the next function. Now, in the group by, it's very important because it doesn't actually go through the entire list when you call the group by. Instead, it will just start going through the list, will give you elements, and it will continue and give you things within the same group for as long as it sees that you have the same value. But if you have a list or some other sequence which is not really sorted by the thing you are grouping by, it means you can get the same group in multiple different places. So let's see an example of that. So say that this time I do not want to group them by the first 2 letters. I want to group them by the GC content. So I create a function that, given a codon, will give me the count of C plus the count of G. So just the number of G's and C's in the codon. Sorry. Now you can see that I get 1 element with 0 count, and then 2 elements with 1, and then another element with 0, and one with 1, and 2 with 2, and so on. So you can see that each one of these numbers actually appears quite many times because it doesn't really sort it. So it just goes for as long as this group continues, and then when it gets to the next group, it will stop. And so it doesn't really guarantee to me that I will get everything with the same GC content together unless I explicitly sort this list by the GC content to begin with. So generally, if you are not sure whether your list is sorted by the thing that you want to group on, it can be very important to actually sort by this value. So for example, suppose that I want to group my VIPERdb records by the genome. I may want to begin by sorting everything by the genome. So I will use a key of itemgetter genome to sort my VIPERdb records, and only then I can use the group by. So in this case, I want to group it by only the first word in the genome, which is the group of each virus. So which would basically divide it to either not available or double-stranded DNA, double-stranded RNA, and all the different groups. And now for each group, I can do any analysis that I want. So for example, I can take this group and take all the families within this record. And by the way, do you know why I use curly brackets here and not square brackets? Not a dictionary? A set, yeah. So it doesn't have a mapping. So it's not a dictionary, it's a set comprehension. So it will give me a set of all the families. And why do I want a set here and not a list? Yeah, I just want the number of families within each group, and there are many records with the same family. I don't want to double count them. So now I can see for each group how many families it has in VIPERdb.